I will begin this video with 7 questions. Please pause the video, copy the questions or try to answer them, make a note of your answers, and I will be explaining the rationale for the correct answer as we go along. The complex immune response results in a protective mechanism which is called immunity. However, sometimes it can also cause a pathogenic condition that is characterized by hypersensitivity. In this Langlumet lithograph, we see le ver solitaire, which is a very big tapeworm that is coming out from this individual who is sick, and the doctor is actually measuring it with a measuring tape. Ragazzo morso d'un ramarro is a painting by Caravaggio from the 16th century, where we can see, I'm going to close up, we can see the lizard there biting the boy. And of course, the boy is responding with pain after this bite. This painting by Edward Munch is a self-portrait of himself with the Spanish flu, painted in the 20th century. What do these 3 paintings or images have in common? Well, what they have in common is that for all of these conditions, our immune system is there to protect us, to provide an immune response that is protective against either the tapeworm or the lizard's venom or the flu virus. Fishing for Oysters at Cancale by Singer Sargent, painted in the 19th century. It is in the permanent collection of the Museum of Fine Arts in Boston. Tres hombres a la mesa, o El almuerzo. It's a painting by Diego Velázquez from the 17th century. And if you see there, these gentlemen are eating what seems to be mussels. So what do these paintings are showing, or why am I showing them together here? Well, in these cases, some individuals may react to shellfish like oysters or mussels or other kind of shellfish with what we know it's actually a pathogenic response of hypersensitivity or allergy to the food. This is not normal. This is not protecting the individual. The terminology of allergy was coined by Clemens von Percat, a pediatrician working in Austria. He correctly identified this reaction as being something that is a hyperreactivity, an abnormal reaction from the immune system that is trying to defend the individual. And the name allergy has 2 components. The first is Greek. It's allos or alien, something that is foreign to the individual. And the second piece, ergy, comes from the German for something that is an action, like energy. So this is an action that the body is taken against something that is alien. His contemporary Charles Richet was also a physician. He was working in France, and he also identified severe abnormal immune reactions, also trying to protect the host. Now, in his case, he saw these more severe and serious cases, and he coined the word anaphylaxis. At first, he called it aphylaxis, which meant not friendly or lacking something that is friendly. But then he thought anaphylaxis would sound better. He did continue his whole career working on what was happening. Why was the immune system reacting so dramatically? And in 1913, he was awarded the Nobel Prize in Medicine and Physiology. Now, these reactions, they are both describing an allergy or an anaphylaxis, and this is what later on will be known as type 1 hypersensitivity reaction. The hypersensitivity described in the 19th century was maintained as type 1 hypersensitivity reaction by the classification that was published in 1963 by Gell and Coombs. In addition, they describe type 2, type 3, and type 4 hypersensitivity reactions. Type 1 is mediated by the antibody IgE and is the one responsible for the atopy, allergy, and anaphylaxis that we are going to be discussing in this particular video. The other hypersensitivity reactions are described in other videos. Déjeuner sur l'herbe is a painting by Claude Monet from the 19th century. Now I like to show this to walk you over some of the concepts of type 1 hypersensitivity, which is when we have allergies to things that we should not be allergic to, like peanuts and other kind of food, allergic to pollens, for instance, allergy to bee stings, or even to pet saliva or dander, which is also in this masterpiece. Type 1 hypersensitivity characterizes allergic and atopic responses. Canestra di frutta is a painting by Caravaggio, and I can immediately perceive that in this fruit basket there's a lot of things that could be allergenic. They could be an allergen, which means they will be identified by antigen-presenting cells, which in turn will bind the T cell with the appropriate co-stimulatory factors that will make this T cell differentiate into what we call Th2 cell that produces interleukin-4, which maturates B cells and allows them to differentiate into plasma cells that will produce IgE. This IgE is going to be specific for that particular antigen. That was the allergen that first came from the fruit basket. Now, the important thing is that this particular IgE that has engaged the allergen will then be identified through the receptor by granulocytes. And when these granulocytes have their receptor with IgE, this produces degranulation. And this degranulation will cause a lot of different effects that end up with a very rapid primary reaction that causes vasodilation, bronchospasm, GI symptoms like diarrhea or vomiting, and skin manifestations like itchiness, like redness, but it also causes endothelial reactions with edema, swelling, and this could be lethal. So although type 1 allergic reactions could be commonly present with sneezing or several different manifestations, they could also be lethal. Ophelia is a painting by John Everett Millais of the Pre-Raphaelite school and is depicting the character in Hamlet who is found dead on the water, surrounded by flowers. And there's always the question of what really killed her. Was she gathering flowers and her dress got wet and she was carried by the waters, or did she jump into the water, or maybe she did have an anaphylactic reaction that caused her death? Immune responses always cause hypersensitivity that can present as an allergic disease. This is wrong because sometimes they actually produce immunity, actually the majority of times. This is the reason why this answer is false. Allergic, atopic, and anaphylactic responses are all classified under the same immediate hypersensitivity response. Yes, this is true. Mi mujer y mis hijas en el jardín from 1910 is a painting by Joaquín Sorolla y Bastida. Now let's talk about what triggers that granulocyte reaction when they are presented with the IgE that has the allergen. So what triggers all of these allergic response? What triggers the allergic response are the allergens, but what are the allergens? Well, they're basically proteins that can be in the environment like pollens, bee stings, peanuts, or pet dandruff. Like you see here, the pet is also depicted in this painting. Now, they could also be chemicals, like in poison ivy, but basically chemicals that modify our own proteins and then make them into allergens. So what are the allergens? They are proteins. Either they are proteins in the environment or there are chemicals in the environment that modify our own proteins. They all have low molecular weight. They're highly soluble, glycosylated. They have enzymatic activity and they are stable. Let's take a look at the key phases that we need to have to mount an allergic or atopic response. The first is loss of tolerance. What does this mean? Well, normally our cells will not be reacting to peanuts or to pollens. We normally can tolerate these when they are being perceived as allergens, when they are perceived as danger signals by the antigen presenting cell or by the epithelium. They will engage the innate lymphoid cells and the Th cells and trigger their response and their differentiation into a Th2 or an ILC2. And these cells are the ones that will be responsible for the next phases of allergic and atopic responses. So the first thing is that these cells are now hyperreacting to an allergen. They lost the tolerance to that allergen. The next thing is this bias of an immune priming to either a Th2 or an ILC2 response. What does this mean? Well, these differentiated cells are going to produce cytokines, and these cytokines are very specific of this phenotype because they are the ones that will create the next level of response. The third phase of the allergic and atopic responses, which is required for these kind of conditions, is activation upon re-exposure. There is memory. This just doesn't happen once. If it happens once and then it disappears, it means that the system became tolerant again. But if you do have an allergic or atopic condition, this just triggers a faster response by the memory B cells that will very quickly be differentiated into plasma cells producing the IgE. And this is what is very characteristic of the allergic and atopic responses. Now, one thing that's important to say is that when we look at the characteristics of this response, we call it type 1 hypersensitivity. Type 2, 3, and 4 hypersensitivity responses were described in 1964 by Gell and Coombs. Type 1 is the one that characterizes allergic responses, and it's called type 1 because it was the first one ever described in the 19th century. However, because the phenotype of the Th cells and the innate lymphocyte cells is the second phenotype, Th2 or ILC2, when we talk about an immune response is type 2. So don't be confused if you're reading clinical documents and you're talking about a clinical response is type 1 hypersensitivity response. If you're looking at an immune response paper, more basic science, and you're talking about an immune response is type 2, but it's the same response. In one case we call it type 1, in the other one we call it type 2, but remember to look at the next words. It's either type 1 hypersensitivity or a type 2 immune response. We're talking about the same. Type 1 hypersensitivity is the same biologic response as type 2 immune response. It is true. They're called differently because they were classified based on different kinds of responses, but we're talking about the same process. Allergies are always triggered by foreign proteins called allergens. This is not true. It can also be caused by endogenous proteins that could become allergens when modified by chemical products. The example I gave is poison ivy. So let me walk you through what is the traditional direct influence of allergen on the dendritic cell and how this model works. Although I'm using the skin as an example, these epithelial cells could be in the lungs, the GI tract, wherever the allergen is in touch with this epithelium, it will be recognized by, in this case, the skin, the dermal dendritic cell, which gets activated and it migrates through the lymphatic system to the secondary lymphoid organs, and it presents to the T cell epitope that stimulates this T cell, having been presented by an allergen, into a Th2 cell. This Th2 cell will start producing interleukin-4 and produce maturation of the B cell. This is still all in the lymph node, but these mature B cells are now proliferate. They travel through the lymph nodes, they drain in the subclavian vein to the circulation, and in the circulation, they differentiate into plasma cells that will produce antibodies. Because it was an allergen epitope and because this was driven by Th2, this is where they produce the IgE antibodies. In addition, these proliferated and differentiated Th2 cells also travel, drain into the circulation, and they will be producing interleukin-5, which stimulates the eosinophils. Now, those dendritic cells that were activated also produce some markers and some migration molecules that take them to the bone marrow, and they will then activate monocytes with the expression of CCR2, which will take them into migration to the site of the allergen exposure. Now, in the contemporary view, in addition to this process, which is still valid, there's an additional component that is important to take into account. When the allergen is exposed to the epithelial and identified as a danger signal, there is activation of that epithelium. That epithelium is not quietly, silently staying in contact with the allergen, but activated. This activation will express a series of different mediators that activate the innate lymphoid cell type 2. And I have other videos that explain both the Th2s and the innate lymphoid cells. These activated innate lymphoid cells produce interleukin-5, which we saw are the ones that will trigger maturation of eosinophils. In addition, they produce also interleukin-13. This interleukin-13 from the ILC2s will stimulate further proliferation of Th2s, but they also, together with interleukin-3, will stimulate and activate macrophages. And all of these processes will produce damage in the epithelium. This is where we see the allergic reactions. And remember that eosinophils are circulating and they are identifying also that damage, and that the IgE is specific to the allergen that was presented to the T cell, at least an epitope of that allergen. So this IgE is now specific and will travel in circulation until it finds where that allergen is. This specific IgE is what characterizes an atopic response. So let's take a look at the role of IgE. The cells that have receptors for IgE are the mast cells and the basophils. As you can see here, I painted the receptor in blue. And this is a high affinity receptor that on exposure to the allergen gets activated. The other cells that have been found to have IgE receptors are the eosinophils, but in this case, this receptor has a lower affinity for the IgE Fc portion. However, it's also specific for that IgE that is bound to the allergen. This activation of the receptors in the cells is going to produce a degranulation. So what is in these granules? What is it that these three types of cells have? Well, the mast cell will have histamine, heparin, chondroitin sulfate, proteases. The basophil has histamine, chondroitin sulfate, several proteases. And the eosinophil has major basic protein, eosinophil cationic protein. Peroxidases, hydrolases, and lipophospholipase. You can see immediately that all of these are very destructive. They are enzymes. So our own cells are producing very nasty products. And why is this happening? Well, remember that the IgE-mediated response is basically dealing with infections from helminths, which are large worms. And to get rid of those, an antibody alone is not sufficient. So these are mechanisms that we have to deal with large organisms that could be pathogenic. And of course, when they are triggered by an allergic response, we are also having this degranulation cause serious adverse reactions. They stimulate several different types of cells in the body. And when it's triggered by IgE, these are usually immediate reactions. This happened within minutes. So that's why when on re-challenge, you get an anaphylactic reaction, you need to observe very quickly. This is going to happen, could be within 5 minutes. On the other hand, we have IgE independent responses, as we saw already, which are part of this type 1 hypersensitivity reaction. The Th2 and ILC2 cells also create memory against those allergens, and they express several different receptors that identify them that produce, instead of granule release, they produce cytokines, typically interleukin-4, interleukin-5, and interleukin-13. These three cytokines bind receptors in a variety of cells, and that's why they cause many different symptoms associated also to allergic responses or type 1 hypersensitivity responses. Now, these are a little bit later because it does take a little longer to get all of these cells proliferating, migrating, relocating to the site of injury or the site where the allergen is. So it can take a little longer than the IgE that's more immediate. The clinical manifestations is what we call an allergic reaction, whereas the specific IgE produced is what we call atopic. So there's a slight difference between the two. They could occur in the same person, but we may have allergic reactions without IgE. Let's take a look at how these cytokines work. So interleukin-4 will bind the cells that bear a receptor, that's a receptor for interleukin-4, which has two chains. Interleukin-4 receptor alpha chain and the gamma chain, the common gamma chain. These intracellularly will activate the Janus kinase system and phosphorylate the stats, producing the appropriate response. It's a type 1 receptor for cytokines. Now, the interleukin-4 receptor alpha chain can also be associated in other cells with the interleukin-13 receptor alpha 1 chain. And this kind of associated receptor can respond to both interleukin-4 and interleukin-13. So cells that bear this kind of receptor will respond to both. They activate and phosphorylate to the Janus kinases and the appropriate stats intracellularly that then produce the response. This is a type 2 receptor. Now, it has been described that interleukin-13 can also bind an interleukin-13 receptor alpha 2, which has no Janus kinases associated intracellularly. And for a long time, this was perceived as potentially a decoy system, something that could dampen a response after some time and will no longer activate cells. However, recently I found that there's some publications that have found that this particular type of receptor eventually produces the tissue growth factor beta and may be associated with fibrosis. So let's take a look at the responses that produce an activation intracellularly. We have interleukin-5 also. The receptor for interleukin-5 has two chains, interleukin-5 receptor alpha and interleukin-5 receptor beta chain. They signal also through activation and phosphorylation of the Janus kinases and STAT1, STAT3, and STAT5, which are present in the eosinophils. This is a very specific eosinophil response, and this will produce eosinophil maturation, survival, and activation. Let's see the role of the basophils in the allergic response. So we know that they have the receptors for IgE and they can degranulate. But there's also an independent of IgE activation of these basophils. And this comes from the contemporary view. When you have an allergen and there's a damage produced on the surface of the cell, we have activation of the epithelium and we have production of several different mediators. Now, TSLP, interleukin 25 and interleukin 33 activate basophils. Even if they're not activated by IgE, they activate these basophils and then they produce interleukin 5, which will target eosinophils. In addition, this basophil that is activated through these mediators will produce interleukin 4 that will produce activation and maturation of B cells with differentiation into plasma cells and production of IgE. This IgE can directly identify the allergen, but remember that it can also activate basophils that have receptors for IgE. And this is what characterizes an atopic response. In addition, the interleukin-4 will stimulate the innate lymphoid cells type 2, which were also activated by those mediators from the epithelium. These activated cells will produce interleukin-5, which further stimulates the eosinophils, but they also produce interleukin-13. That's also a product of basophil activation, and they both will stimulate further proliferation of Th2 responses. So it is complex. It goes many different ways, and there's a lot of interaction, but we still see these three cytokines, interleukin-4, interleukin-5, interleukin-13, as being critical in the allergic response. So this is what's being called TSLP-mediated activation, because this begins with that contemporary hypothesis of allergic reactions that will not be dependent on IgE. And the other activation of basophils is IgE-mediated activation. Now remember that TSLP could lead eventually to plasma cell and IgE production, so there may be a mix. And this is where I found some publications that mention that there's a little bit of a different balance between different diseases, whereas food allergy will be more predominantly IgE-mediated activation with the basophil degranulation, all the way to eosinophilic esophagitis, which, according to these investigators, could be more typical of a TSLP-mediated activation of the basophils. The eosinophils have IgE receptor, but in allergic responses, they can also have a reaction to interleukin-5. The interleukin-5 receptor, which is present in the eosinophils, and to my knowledge, no other cells have been found to have this receptor, will signal through two chains in the receptor, interleukin-5 receptor alpha and interleukin-5 receptor beta, using the Janus kinase system and activating STAT1, STAT3, and STAT5. This will create eosinophil maturation, survival, and activation. It's a key cytokine for eosinophils. Now, on the other hand, interleukin-4, which is also driven by the hypersensitivity type 1 response or type 2 immune response, will maturate B cells that differentiate into plasma cells producing IgE, and this IgE will also engage the other receptors in the eosinophils. So eosinophils have receptors for interleukin-5, and they also have receptors for IgE. The function of these two receptors is very different. When interleukin-5 is engaging the eosinophil receptor, it produces maturation, survival, and activation of the eosinophil population. But when the IgE bound to an allergen is engaging that receptor in the eosinophil, it produces degranulation. And this releases a lot of different proteins that will destroy a lot of the cell types around it. Remember, this is a normal response against worms, against helminthic infections. These are very large organisms that can produce diseases in the organism. And that's the reason why these granules contain these very potent proteins that destroy the other cells, the cells that are of the helminthic infection. Of course, in conditions of atopic or allergic diseases, they are destroying our own epithelium. The key cells that respond to IgE are called granulocytes, which is true, and include only basophils and eosinophils. No, that's not true. Mast cells also contain the receptors for IgE and are involved in these responses. IL-4, IL-5, and IL-13 characterize type 2 immune response. This is true. There are many other cytokines, but these three characterize the type 2 immune response and the consequent type 1 hypersensitivity reaction. TSLP and interleukin-33 are among other cytokines that can stimulate basophils via the IgE receptor. That's not true. This is independent of the IgE receptor. It's via their corresponding receptors. Thank you very much for watching this video. I hope you found it informative, and I hope that you learned something from watching it. Please remember that I have other videos in my channel, also educational videos, and consider subscribing to my channel. Thank you very much and have a good day.